Here we'll look at the efficiency of absorption. It means when we're looking at absorption and the way it occurs, we ask ourselves, what structural suitabilities does the small intestine, because that's where absorption mostly occurs, what structural suitabilities, or how is the small intestine structured suitably such that it can ensure that absorption can occur in the most efficient way? That's what we are trying now to investigate. And our first point here, we're looking at the length of the small intestine itself. The small intestine is not short, it is actually very long. In human adults, it can reach lengths of up to 6 meters. That's quite long, longer than any human being. Now, what is the purpose of having that kind of length? It ensures that the food will remain for a long period of time in the alimentary canal. This is to allow enough time for absorption to take place. Or imagine there's a lot of things passing on a trail that's very long, it's continuous. If that trail is very long and is passing in front of you, you will then have time to pick up as much as you can from the trail if it's carrying a lot of material. But if it's just a short one and it's passing in front of you, you don't have enough time to pick up anything. Everything just drops down on the ground. So with a longer trail, you have enough time to pick up as much as you want. And we are saying pick up, it actually means absorbing. So there's enough time to absorb because of the length of the small intestine. Secondly, when we look at the small intestine, we see the presence of finger-like projections. These are the villi. There are millions of them that are found inside the intestine, in the intestinal walls. These also increase the surface area for absorption because the villi absorb nutrients through their surface, through their wall. Means the presence of so many villi will mean each villus will be there doing absorption all around its surface, and other villi will be doing the same thing. At the same time, so many villi present, then they will actively absorb nutrients much more efficiently. Again, when we look at the wall of the villus, it is actually thin. And being thin, it makes sure that things material, nutrients like amino acids, for example, can easily pass through. And if they can easily pass through to go to the other side, into the villus, into the capillaries or the lacteals, then material can easily be absorbed. Imagine that the wall of the villus was thick. It means we can't easily pass through that. Okay, this will prevent nutrients from getting easy access to the blood capillaries or the lymph, where they have to be distributed all around, from where they have to be distributed all to all parts of the body. So a thin wall ensures that absorption occurs very efficiently. And again, it has this small intestine, or the villus, has a large supply of lacteals and capillaries. What are these meant for? Lacteals are there to transport fats, that's fatty acid and glycerol. Capillaries are there to transport glucose and amino acids. It means there's already a transport system that is available within the villi or within the small intestine to make sure that whatever needs to be absorbed gets absorbed and gets transported very quickly. Imagine you have to transport so many people at once. What will be better? What will make your job much more easy? Is it the availability of so many vehicles or the availability of just few vehicles? I'm sure if you have few vehicles, then you will transport less people at a time. But if you have so many vehicles, then you will transport very fast and very efficiently. And that's what we are saying here. A large supply of these vessels, the lacteals and capillaries, it means a large supply of transport system of vehicles that are there to transport what will be absorbed. It then means that when we have a lot of capillaries and lacteals, then absorption can be done very quickly. Again, when we look at the columnar epithelial cells that are found in the walls of the villi, we will notice that there is also the presence of microvilli. It means each villus has its own small, many villi around it itself. So these secondary villi are smaller, and hence we call them microvilli, which are present on the surface of the villi themselves or on the walls of the columnar epithelial cells of the villi. And they are there. Each of these microvilli does the same thing as the real big villi does. They are also there to absorb. So each one is participating in this absorption. At the end of the day, we have a large surface area for absorption. Again, within the columnar epithelial cells of the villi, because they are cells like ordinary cells, they have a higher concentration of mitochondria. And mitochondria among the organelles are referred to as the powerhouse of the cell because they are the site for manufacturing energy. But this energy will be used for what? The energy will then be used to facilitate active transport that we said always requires energy. So with a large amount of mitochondria, we are then guaranteed that if substances need to be transported actively, then there will be enough energy to support that requirement in energy for active transport. Hence, a large number of mitochondria will be present at the cells or in the cells of the columnar epithelia of the villi. So these are the points that facilitate absorption. Transport and assimilation. In this case of transport, we're looking at how the nutrients, the breakdown products of digestion, are transported to various parts of the body and distributed to the various cells. And in assimilation, this involves the taking in of these soluble, simpler substances, nutrients, by various cells of the body. So how does transport and assimilation occur? 3 stages. 1, 2, and 3. Okay. The first part of it involves glucose, amino acids, vitamins, ions, and water. How are they transported and assimilated? From the mucosa of the small intestine. Now, from the mucosa of the small intestine, these substances enter the blood capillaries of the villi. We've seen that. Only fat so far was going into the lactin, as far as we've covered. To enter the blood capillaries, here are the blood capillaries, and this is the villus. So we're looking here at amino acids and glucose as part of these substances I've been mentioning up here. So that's the villus. These substances have entered the blood capillaries of the villus, and that's from there that we're going to see how they travel. From the blood capillaries of the villus, these substances will then pass through the hepatic portal vein, and that hepatic portal vein is then going to carry the substances to the liver. And in the liver, some other processes will occur. First, in the liver, the excess glucose that was transported by active transport will then be converted to glycogen. Glycogen in animals is a storage form of polysaccharides. The same way as plant stores polysaccharide in the form of starch, animals store polysaccharides in the form of glycogen. And how are these polysaccharides made? They are made of simple units of sugars. It means you put one glucose with another glucose with another one. Many, poly means many. Many subunits of sugar combined together will give a polysaccharide, and it will then be stored in animals in the form of glycogen, in plants in the form of starch. And that's why we had to break down starch using salivary amylase in the mouth so that we can form glucose and later on recombine it to form our own form of polysaccharide, which is glycogen. This will be stored in the liver. Again, excess amino acid that also went through the hepatic portal vein to the liver. Excess amino acid will also be deaminated. It will be broken down to form urea or uric acid, which will then be excreted by the kidneys. We'll see that when we are doing excretion. Now the remaining glucose and amino acids, remember we are here just either converting excess glucose or breaking down amino acids. All what we were dealing with was the excess. The remaining useful ones, remaining useful glucose and amino acid, will then leave the liver, passing through the hepatic vein. Please don't confuse this with the hepatic portal vein. The hepatic portal vein takes from the capillaries of the mucosa of the villus and through the hepatic portal vein to the liver. Then from the liver, it will then pass through the hepatic vein and it will then reach the heart, because that is the major organ that's going to distribute all these nutrients. It will then reach the heart through a lower blood vessel that we call the inferior vena cava, that takes all these amino acids to the heart. We'll talk about what happens from there. It enters through the heart, through the right side of the heart. That's where the vena cava, there are two of them. There's the inferior and there is the superior. That's where they make contact with the heart, to the right side of the heart. So we'll look at what happens from that point. On the other hand, fatty acids and glycerol, remember, they are taken up by the lacteals of the small villi. Here are fatty acids and glycerol being taken up by the lacteals of the villus. Now, from there, they will be transported, because the lacteals will merge. It's just like capillaries merging to form vein or arteries. The lacteals also merge to form larger vessels, which are called lymph vessels. And the fatty acids and glycerol will then be transported by these lymph vessels to the left thoracic duct, found around the thorax. Will be transported to the left thoracic duct, and the left thoracic duct will in turn transport this fatty acid and glycerol and empty them into the blood system, into the veins, such that they meet up with blood coming from the head, blood coming from the hand, and everything will be delivered now to the superior vena cava. So that's the point where we are at. We are at 2 points here. I want you to realize 2 points where we are. We have here the inferior vena cava that brought in amino acids and glucose to the right side of the heart. We are now here with the superior vena cava that's bringing in fatty acid and glycerol also to the right side of the heart. Now, from the right side of the heart, where will this go to? Here is where we are now. Number 3. That's where we are at point number 3. From the right side of the eye of the heart, where will all of these go to? The heart will then have to pump them, pump the blood. At this point, the blood here contains amino acids, it contains glucose, it contains fatty acids and glycerol. The heart will then pump everything passing through the pulmonary artery. And from the pulmonary artery, it's then going to go to the lungs. That's where all the blood will go to the lungs. It means this blood will have amino acids, fatty acids, and glycerol and glucose. All of them, everything with the vitamins included as well. Everything will go to the lungs to get oxygenated, because at this point, most of this blood which came from all the parts of the body had already lost their oxygen. That is why blood has to be pumped through the pulmonary artery to go first to the lungs to go and transport oxygen, so that by the time the blood leaves the lungs and is coming back to the heart, this blood which is leaving the lungs and coming back here to the heart, coming down to the left side of the heart, passing through the pulmonary vein, that blood now contains all these nutrients, the amino acids, etc., plus oxygen. That's why we also refer to it as oxygenated blood. And this is now blood which is rich, the kind of blood that the cells will want to get so that they can get nutrients, they can get oxygen as well. This blood now will be pumped from the left side of the heart, passing through the aorta. And the aorta now pumps the blood or sends the blood through to all parts of the body, and the cycle can start all over again, because when it goes to all parts of the body, it means it also comes back to the small intestine, where it will go again into the villus, which will then again transport amino acids. It will go through the lacteals, which will also transport fatty acids and glycerol, and the capillaries have amino acids and glucose, and the cycle continues, and everything comes back here to the heart, and then it pumps again, and that's how it happens. Now, assimilation then. Assimilation will then occur such that nutrients are being absorbed, because when the heart pumps the blood, which is laden with oxygen and nutrients, to the rest of the body from the aorta, it goes now to the individual cells at every part of the body you can think about. Is it the eye, the toes, the nose, the tongue? Every part, the fingers, everywhere. Every cells of those parts will receive this blood which contains all of these nutrients together with oxygen. And these nutrients will then move into the cell by diffusion. They move into the cell. That movement, or going into the cell, that absorption, is what we refer to as assimilation. They are being assimilated, passing through the cell membranes, because remember, the cell membranes are semi-permeable. They have pores through which material can pass, and the cells assimilate, and that is how now the cells also receive food, so that they can use these nutrients to build cellular structures, like again the cell membrane itself. That's why it's made up of a bilayer of phospholipid, that's fat and proteins, like the cell inclusions or the cytoplasmic inclusions, other organelles like the mitochondria, the Golgi body, and the plasmic reticulum, and you can name the rest. So all of these nutrients are used for building these cellular structures and for making proteins in the body that will either be functional or structural. That's the way the body gets nutrients back to itself.